Epigenetics.
In biology, and specifically genetics, epigenetics is the study of heritable changes in gene activity that are not caused by changes in the DNA sequence. It also can be used to describe the study of stable, long-term alterations in the transcriptional potential of a cell that are not necessarily heritable. Unlike simple genetics based on changes to the DNA sequence, the changes in gene expression or cellular phenotype of epigenetics have other causes, thus use of the term epigenetics. The term also refers to the changes themselves, functionally relevant changes to the genome that do not involve a change in the nucleotide sequence. Examples of mechanisms that produce such changes are DNA methylation and histone modification, each of which alters how genes are expressed without altering the underlying DNA sequence. Gene expression can be controlled through the action of repressor proteins that attach to silencer regions of the DNA. These epigenetic changes may last through cell divisions for the duration of the cell's life, and may also last for multiple generations even though they do not involve changes in the underlying DNA sequence of the organism. Instead, non-genetic factors cause the organism's genes to behave, or express themselves differently. There are objections to the use of the term epigenetic to describe chemical modification of histone, since it remains unclear whether or not histone modifications are heritable. One example of an epigenetic change in eukaryotic biology is the process of cellular differentiation. During morphogenesis, totipotent stem cells become the various pluripotent cell lines of the embryo, which in turn become fully differentiated cells. In other words, as a single fertilized egg cell, the zygote, continues to divide, the resulting daughter cells change into all the different cell types in an organism, including neurons, muscle cells, epithelium, endothelium of blood vessels, etc., by activating some genes while inhibiting the expression of others. In 2011, it was demonstrated that the methylation of mRNA plays a critical role in human energy homeostases. The obesity-associated FTO gene is shown to be able to demethylate N6-methyladenosine in RNA. This discovery launched the subfield of RNA epigenetics.
Historical usage of epigenetics.
Epigenetics, as in epigenetic landscape, was coined by C. H. Waddington in 1942 as a portmanteau of the words epigenesis and genetics. Epigenesis is an old word that has more recently been used, see proformationism for historical background, to describe the differentiation of cells from their initial totipotent state in embryonic development. When Waddington coined the term, the physical nature of genes and their role in heredity was not known. He used it as a conceptual model of how genes might interact with their surroundings to produce a phenotype. He used the phrase epigenetic landscape as a metaphor for biological development. Waddington held that cell fates were established in development much like a marble rolls down to the point of lowest local elevation. Waddington suggested visualizing increasing irreversibility of cell type differentiation as ridges rising between the valleys where the marbles, cells, are traveling. In recent times, Waddington's notion of the epigenetic landscape has been rigorously formalized in the context of the systems dynamics state approach to the study of cell fate. The term epigenetics has also been used in developmental psychology to describe psychological development as the result of an ongoing, bidirectional interchange between heredity and the environment. Interactivist ideas of development have been discussed in various forms and under various names throughout the 19th and 20th centuries. An early version was proposed, among the founding statements in embryology, by Carl Ernst von Baer and popularized by Ernst Haeckel. A radical epigenetic view, physiological epigenesis, was developed by Paul Winter-Burt. Another variation, probabilistic epigenesis, was presented by Gilbert Gottlieb in 2003. This view encompasses all of the possible developing factors on an organism and how they not only influence the organism and each other but how the organism also influences its own development. Noted developmental psychologist Erik Erikson used the term epigenetic principle in his book Identity, Youth and Crisis, 1968, and used it to encompass the notion that we develop through an unfolding of our personality in predetermined stages, and that our environment and surrounding culture influence how we progress through these stages. This biological unfolding in relation to our socio-cultural settings is done in stages of psychosocial development, where progress through each stage is in part determined by our success, or lack of success, in all the previous stages.
Contemporary usage of epigenetics.
Robin Holliday defined epigenetics as the study of the mechanisms of temporal and spatial control of gene activity during the development of complex organisms. Thus epigenetic can be used to describe anything other than DNA sequence that influences the development of an organism. The more recent usage of the word in science has a stricter definition. It is, as defined by Arthur Riggs and colleagues, the study of mitotically and or meiotically heritable changes in gene function that cannot be explained by changes in DNA sequence. The Greek prefix epi in epigenetics implies features that are on top of, or in addition to genetics. Thus epigenetic traits exist on top of or in addition to the traditional molecular basis for inheritance. The term epigenetics, however, has been used to describe processes which have not been demonstrated to be heritable, such as histone modification. There are therefore attempts to redefine it in broader terms that would avoid the constraints of requiring heritability. For example, Sir Adrian Bird defined epigenetics as the structural adaptation of chromosomal regions so as to register, signal or perpetuate altered activity states. This definition would be inclusive of transient modifications associated with DNA repair or cell cycle phases as well as stable changes maintained across multiple cell generations, but exclude others such as templating of membrane architecture and prions unless they impinge on chromosome function. Such redefinitions however are not universally accepted and are still subject to dispute. The NIH Roadmap Epigenomics Project, ongoing as of 2013, uses the following definition: epigenetics is an emerging frontier of science that involves the study of changes in the regulation of gene activity and expression that are not dependent on gene sequence. For purposes of this program, epigenetics refers to both heritable changes in gene activity and expression, in the progeny of cells or of individuals, and also stable, long-term alterations in the transcriptional potential of a cell that are not necessarily heritable. While epigenetics refers to the study of single genes or sets of genes, epigenomics refers to more global analyses of epigenetic changes across the entire genome. In 2008, a consensus definition of the epigenetic trait, stably heritable phenotype resulting from changes in a chromosome without alterations in the DNA sequence, was made at a Cold Spring Harbor meeting. The similarity of the word to genetics has generated many parallel usages. The epigenome is a parallel to the word genome, and refers to the overall epigenetic state of a cell. The phrase genetic code has also been adapted. The epigenetic code has been used to describe the set of epigenetic features that create different phenotypes in different cells. Taken to its extreme, the epigenetic code could represent the total state of the cell, with the position of each molecule accounted for in an epigenomic map, a diagrammatic representation of the gene expression, DNA methylation and histone modification status of a particular genomic region. More typically, the term is used in reference to systematic efforts to measure specific, relevant forms of epigenetic information such as the histone code or DNA methylation patterns.
Molecular basis of epigenetics.
Epigenetic changes can modify the activation of certain genes, but not the sequence of DNA. Additionally, the chromatin proteins associated with DNA may be activated or silenced. This is why the differentiated cells in a multicellular organism express only the genes that are necessary for their own activity. Epigenetic changes are preserved when cells divide. Most epigenetic changes only occur within the course of one individual organism's lifetime, but, if gene inactivation occurs in a sperm or egg cell that results in fertilization, then some epigenetic changes can be transferred to the next generation. This raises the question of whether or not epigenetic changes in an organism can alter the basic structure of its DNA, see Evolution, below, a form of Lamarckism. Specific epigenetic processes include paramutation, bookmarking, imprinting, gene silencing, X chromosome inactivation, position effect, reprogramming, transfection, maternal effects, the progress of carcinogenesis, many effects of teratogens, regulation of histone modifications and heterochromatin, and technical limitations affecting parthenogenesis and cloning. DNA damage can also cause epigenetic changes. DNA damages are very frequent, occurring on average about 10,000 times a day per cell of the human body, see DNA damage, naturally occurring. These damages are largely repaired, but at the site of a DNA repair, epigenetic changes can remain. In particular, a double-strand break in DNA can initiate unprogrammed epigenetic gene silencing both by causing DNA methylation as well as by promoting silencing types of histone modifications, chromatin remodeling, see next section. In addition, the enzyme PARP1, poly, ADP, ribose polymerase, and its product poly, ADP, ribose, PAR, accumulate at sites of DNA damage as part of a repair process. This accumulation, in turn, directs recruitment and activation of the chromatin remodeling protein ALC1 that can cause nucleosome remodeling. Nucleosome remodeling has been found to cause, for instance, epigenetic silencing of DNA repair gene MLH1. DNA-damaging chemicals, such as benzene, hydroquinone, styrene, carbon tetrachloride and trichloroethylene, cause considerable hypermethylation of DNA, some through the activation of oxidative stress pathways. Foods are known to alter the epigenetics of rats on different diets. Some food components epigenetically increase the levels of DNA repair enzymes such as management and MLH1 and P53. Other food components can reduce DNA damage, such as soy isoflavones and bilberry anthocyanins. Epigenetic research uses a wide range of molecular biologic techniques to further our understanding of epigenetic phenomena, including chromatin immunoprecipitation, together with its large-scale variants chip-on-chip and chip-seq, fluorescent in situ hybridization, methylation-sensitive restriction enzymes, DNA adenine methyltransferase identification, DAMID, and bisulfite sequencing. Furthermore, the use of bioinformatic methods is playing an increasing role, computational epigenetics. Computer simulations and molecular dynamics approaches revealed the atomistic motions associated with the molecular recognition of the histone tail through an allostric mechanism.
Mechanisms.
Several types of epigenetic inheritance systems may play a role in what has become known as cell memory. Note however that not all of these are universally accepted to be examples of epigenetics.
DNA methylation and chromatin remodeling.
Because the phenotype of a cell or individual is affected by which of its genes are transcribed, heritable transcription states can give rise to epigenetic effects. There are several layers of regulation of gene expression. One way that genes are regulated is through the remodeling of chromatin. Chromatin is the complex of DNA and the histone proteins with which it associates. Histone proteins are little spheres that DNA wraps around. If the way that DNA is wrapped around the histones changes, gene expression can change as well. Chromatin remodeling is accomplished through two main mechanisms. The first way is post-translational modification of the amino acids that make up histone proteins. Histone proteins are made up of long chains of amino acids. If the amino acids that are in the chain are changed, the shape of the histone sphere might be modified. DNA is not completely unwound during replication. It is possible, then, that the modified histones may be carried into each new copy of the DNA. Once there, these histones may act as templates, initiating the surrounding new histones to be shaped in the new manner. By altering the shape of the histones around it, these modified histones would ensure that a differentiated cell would stay differentiated, and not convert back into being a stem cell. The second way is the addition of methyl groups to the DNA, mostly at CpG sites, to convert cytosine to 5-methylcytosine. 5-methylcytosine performs much like a regular cytosine, pairing up with a guanine. However, some areas of the genome are methylated more heavily than others, and highly methylated areas tend to be less transcriptionally active, through a mechanism not fully understood. Methylation of cytosines can also persist from the germline of one of the parents into the zygote, marking the chromosome as being inherited from this parent, genetic imprinting. The way that the cells stay differentiated in the case of DNA methylation is clearer to us than it is in the case of histone shape. Basically, certain enzymes, such as DNMT1, have a higher affinity for the methylated cytosine. If this enzyme reaches a hemimethylated portion of DNA, where methylcytosine is in only one of the two DNA strands, the enzyme will methylate the other half. Although histone modifications occur throughout the entire sequence, the unstructured N-termini of histones, called histone tails, are particularly highly modified. These modifications include acetylation, methylation, ubiquitylation, phosphorylation, sumylation, ribosylation and citrullination. Acetylation is the most highly studied of these modifications. For example, acetylation of the K14 and K9 lysines of the tail of histone H3 by histone acetyltransferase enzymes, HATs, is generally correlated with transcriptional competence. One mode of thinking is that this tendency of acetylation to be associated with active transcription is biophysical in nature. Because it normally has a positively charged nitrogen at its end, lysine can bind the negatively charged phosphates of the DNA backbone. The acetylation event converts the positively charged N group on the side chain into a neutral amide linkage. This removes the positive charge, thus loosening the DNA from the histone. When this occurs, complexes like SWISNF and other transcriptional factors can bind to the DNA and allow transcription to occur. This is the Psi model of epigenetic function. In other words, changes to the histone tails have a direct effect on the DNA itself. Another model of epigenetic function is the trans model. In this model, changes to the histone tails act indirectly on the DNA. For example, lysine acetylation may create a binding site for chromatin-modifying enzymes, and basal transcription machinery as well. This chromatin remodeler can then cause changes to the state of the chromatin. Indeed, the bromodomain, a protein segment, domain, that specifically binds acetylysine, is found in many enzymes that help activate transcription, including the SWISNF complex, on the protein polybromo. It may be that acetylation acts in this and the previous way to aid in transcriptional activation. The idea that modifications act as docking modules for related factors is borne out by histone methylation as well. Methylation of lysine 9 of histone H3 has long been associated with constitutively transcriptionally silent chromatin, constitutive heterochromatin. It has been determined that a chromodomain, a domain that specifically binds methyl lysine, in the transcriptionally repressive protein HP1 recruits HP1 to K9 methylated regions. One example that seems to refute this biophysical model for methylation is that trimethylation of histone H3 at lysine 4 is strongly associated with, and required for full, transcriptional activation. Trimethylation in this case would introduce a fixed positive charge on the tail. It has been shown that the histone lysine methyltransferase, KMT, is responsible for this methylation activity in the pattern of histones H3 and H4. This enzyme utilizes a catalytically active site called the set domain, suppressor of variegation, enhancer of zest, triathorax. The set domain is a 130 amino acid sequence involved in modulating gene activities. This domain has been demonstrated to bind to the histone tail and causes the methylation of the histone. Differing histone modifications are likely to function in differing ways. Acetylation at one position is likely to function differently from acetylation at another position. Also, multiple modifications may occur at the same time, and these modifications may work together to change the behavior of the nucleosome. The idea that multiple dynamic modifications regulate gene transcription in a systematic and reproducible way is called the histone code. DNA methylation frequently occurs in repeated sequences, and helps to suppress the expression and mobility of transposable elements because 5-methylcytosine can be spontaneously deaminated, replacing nitrogen by oxygen, to thymidine. CpG sites are frequently mutated and become rare in the genome, except at CpG islands where they remain unmethylated. Epigenetic changes of this type thus have the potential to direct increased frequencies of permanent genetic mutation. DNA methylation patterns are known to be established and modified in response to environmental factors by a complex interplay of at least 3 independent DNA methyltransferases, DNMT1, DNMT3A, and DNMT3B, the loss of any of which is lethal in mice. DNMT1 is the most abundant methyltransferase in somatic cells, localizes to replication foci, has a 10-40-fold preference for hemimethylated DNA and interacts with the proliferating cell nuclear antigen, pCNA. By preferentially modifying hemimethylated DNA, DNMT1 transfers patterns of methylation to a newly synthesized strand after DNA replication, and therefore is often referred to as the maintenance methyltransferase. DNMT1 is essential for proper embryonic development, imprinting and exon activation. To emphasize the difference of this molecular mechanism of inheritance from the canonical Watson-Crick base pairing mechanism of transmission of genetic information, the term epigenetic templating was introduced. Furthermore, in addition to the maintenance and transmission of methylated DNA states, the same principle could work in the maintenance and transmission of histone modifications and even cytoplasmic, structural, heritable states. Histones H3 and H4 can also be manipulated through demethylation using histone lysine demethylase, KDM. This recently identified enzyme has a catalytically active site called the Umunji domain, JMJC. The demethylation occurs when JMJC utilizes multiple cofactors to hydroxylate the methyl group, thereby removing it. JMJC is capable of demethylating mono, D, and trimethylated substrates. Chromosomal regions can adopt stable and heritable alternative states resulting in bistable gene expression without changes to the DNA sequence. Epigenetic control is often associated with alternative covalent modifications of histones. The stability and heritability of states of larger chromosomal regions are often thought to involve positive feedback where modified nucleosomes recruit enzymes that similarly modify nearby nucleosomes. A simplified stochastic model for this type of epigenetics is found here. Because DNA methylation and chromatin remodeling plays such a central role in many types of epigenetic inheritance, the word epigenetics is sometimes used as a synonym for these processes. However, this can be misleading. Chromatin remodeling is not always inherited, and not all epigenetic inheritance involves chromatin remodeling. It has been suggested that the histone code could be mediated by the effect of small RNAs. The recent discovery and characterization of a vast array of small, 21 to 26 nt, non-coding RNAs suggests that there is an RNA component, possibly involved in epigenetic gene regulation. Small interfering RNAs can modulate transcriptional gene expression via epigenetic modulation of targeted promoters.
RNA transcripts and their encoded proteins.
Sometimes a gene, after being turned on, transcribes a product that, either directly or indirectly, maintains the activity of that gene. For example, HNF4 and MyoD enhance the transcription of many liver and muscle-specific genes, respectively, including their own, through the transcription factor activity of the proteins they encode. RNA signaling includes differential recruitment of a hierarchy of generic chromatin-modifying complexes and DNA methyltransferases to specific loci by RNAs during differentiation and development. Other epigenetic changes are mediated by the production of different splice forms of RNA, or by formation of double-stranded RNA, RNAi. Descendants of the cell in which the gene was turned on will inherit this activity, even if the original stimulus for gene activation is no longer present. These genes are most often turned on or off by signal transduction, although in some systems where syncytia or gap junctions are important, RNA may spread directly to other cells or nuclei by diffusion. A large amount of RNA and protein is contributed to the zygote by the mother during oogenesis or via NER cells, resulting in maternal effect phenotypes. A smaller quantity of sperm RNA is transmitted from the father, but there is recent evidence that this epigenetic information can lead to visible changes in several generations of offspring.
MicroRNAs.
MicroRNAs, mRNAs, are members of non-coding RNAs that range in size from 17 to 25 nucleotides. As indicated by Wang et al., mRNAs regulate a large variety of biological functions in plants and animals. So far, in 2013, about 2,000 mRNAs have been discovered in humans, and these can be found online in an mRNA database. Each mRNA expressed in a cell may target about 100 to 200 messenger RNAs that it downregulates. Most of the downregulation of mRNAs occurs by causing the decay of the targeted mRNA, while some downregulation occurs at the level of translation into protein. It appears that about 60% of human protein coding genes are regulated by mRNAs. Many mRNAs are epigenetically regulated. About 50% of mRNA genes are associated with CpG islands, that may be repressed by epigenetic methylation. Transcription from methylated CpG islands is strongly and heritably repressed. Other mRNAs are epigenetically regulated by either histone modifications or by combined DNA methylation and histone modification.
sRNAs.
sRNAs are small, 50 to 250 nucleotides, highly structured, non-coding RNA fragments found in bacteria. They control gene expression including virulence genes in pathogens and are viewed as new targets in the fight against drug-resistant bacteria. They play an important role in many biological processes, binding to mRNA and protein targets in prokaryotes. Their phylogenetic analyses, for example through sRNA-mRNA target interactions or protein binding properties, are used to build comprehensive databases. sRNA gene maps based on their targets in microbial genomes are also constructed.
Prions.
Prions are infectious forms of proteins. In general, proteins fold into discrete units that perform distinct cellular functions, but some proteins are also capable of forming an infectious conformational state known as a prion. Although often viewed in the context of infectious disease, prions are more loosely defined by their ability to catalytically convert other native state versions of the same protein to an infectious conformational state. It is in this latter sense that they can be viewed as epigenetic agents capable of inducing a phenotypic change without a modification of the genome. Fungal prions are considered by some to be epigenetic because the infectious phenotype caused by the prion can be inherited without modification of the genome. PSI+ and URE3, discovered in yeast in 1965 and 1971, are the two best studied of this type of prion. Prions can have a phenotypic effect through the sequestration of protein in aggregates, thereby reducing that protein's activity. In PSI+ cells, the loss of the sub-35 protein, which is involved in termination of translation, causes ribosomes to have a higher rate of read-through of stop codons, an effect that results in suppression of nonsense mutations in other genes. The ability of sub-35 to form prions may be a conserved trait. It could confer an adaptive advantage by giving cells the ability to switch into a PSI+ state and express dormant genetic features normally terminated by stop codon mutations.
Structural inheritance systems.
In ciliates such as tetraamina and paramecium, genetically identical cells show heritable differences in the patterns of ciliary rows on their cell surface. Experimentally altered patterns can be transmitted to daughter cells. It seems existing structures act as templates for new structures. The mechanisms of such inheritance are unclear, but reasons exist to assume that multicellular organisms also use existing cell structures to assemble new ones.
Functions and consequences. Development.
Somatic epigenetic inheritance through epigenetic modifications, particularly through DNA methylation and chromatin remodeling, is very important in the development of multicellular eukaryotic organisms. The genome sequence is static, with some notable exceptions, but cells differentiate into many different types, which perform different functions, and respond differently to the environment and intercellular signaling. Thus, as individuals develop, morphogens activate or silence genes in an epigenetically heritable fashion, giving cells a memory. In mammals, most cells terminally differentiate, with only stem cells retaining the ability to differentiate into several cell types, totipotency, and multipotency. In mammals, some stem cells continue producing new differentiated cells throughout life, such as in neurogenesis, but mammals are not able to respond to loss of some tissues, for example, the inability to regenerate limbs, which some other animals are capable of. Unlike animals, plant cells do not terminally differentiate, remaining totipotent with the ability to give rise to a new individual plant. While plants do utilize many of the same epigenetic mechanisms as animals, such as chromatin remodeling, it has been hypothesized that some kinds of plant cells do not use or require cellular memories, resetting their gene expression patterns using positional information from the environment and surrounding cells to determine their fate. Epigenetics can be divided into predetermined and probabilistic epigenesis. Predetermined epigenesis is a unidirectional movement from structural development in DNA to the functional maturation of the protein. Predetermined here means that development is scripted and predictable. Probabilistic epigenesis, on the other hand, is a bidirectional structure-function development with experiences and external molding development.
Medicine.
Epigenetics has many and varied potential medical applications as it tends to be multidimensional in nature. Congenital genetic disease is well understood, and it is also clear that epigenetics can play a role, for example, in the case of Angelman syndrome and Prader-Willi syndrome. These are normal genetic diseases caused by gene deletions or inactivation of the genes, but are unusually common because individuals are essentially homozygous because of genomic imprinting, and therefore a single gene knockout is sufficient to cause the disease, where most cases would require both copies to be knocked out.
Evolution.
Epigenetics can impact evolution when epigenetic changes are heritable. A sequestered germline or Weissman barrier is specific to animals, and epigenetic inheritance is more common in plants and microbes. Eva Jablonka and Marion Lamb have argued that these effects may require enhancements to the standard conceptual framework of the modern evolutionary synthesis. Other evolutionary biologists have incorporated epigenetic inheritance into population genetics models or are openly skeptical. Two important ways in which epigenetic inheritance can be different from traditional genetic inheritance, with important consequences for evolution, are that rates of EFI mutation can be much faster than rates of mutation and the EFI mutations are more easily reversible. An epigenetically inherited element such as the PSI plus system can act as a stopgap, good enough for short-term adaptation that allows the lineage to survive for long enough for mutation and or recombination to genetically assimilate the adaptive phenotypic change. The existence of this possibility increases the evolvability of a species.
Current research findings and examples of effects.
Epigenetic changes have been observed to occur in response to environmental exposure. For example, mice given some dietary supplements have epigenetic changes affecting expression of the agouti gene, which affects their fur color, weight, and propensity to develop cancer. One study indicates that traumatic experiences can produce fearful memories which are passed to future generations via epigenetics. A study carried out on mice in 2013 found that mice could produce offspring which had an aversion to certain items which had been the source of negative experiences for their ancestors. Reports stated that in the case of humans with different environmental exposures, Frigo et al. Studied young monozygotic, identical, twins and older monozygotic twins. They found that although such twins were epigenetically indistinguishable during their early years, Older twins had remarkable differences in the overall content and genomic distribution of 5-methylcytosine DNA and histone acetylation. The twin pairs who had spent less of their lifetime together and/or had greater differences in their medical histories were those who showed the largest differences in their levels of 5-methylcytosine DNA and acetylation of histones H3 and H4. More than 100 cases of transgenerational epigenetic inheritance phenomena have been reported in a wide range of organisms, including prokaryotes, plants, and animals. For instance, mourning cloak butterflies will change color through hormone changes in response to experimentation of varying temperatures. Recent analyses have suggested that members of the Apibec family of cytosine deaminases are capable of simultaneously mediating genetic and epigenetic inheritance using similar molecular mechanisms.
Epigenetic effects in humans. Genomic imprinting and related disorders.
Some human disorders are associated with genomic imprinting, a phenomenon in mammals where the father and mother contribute different epigenetic patterns for specific genomic loci in their germ cells. The best-known case of imprinting in human disorders is that of Angelman syndrome and Prader-Willi syndrome. Both can be produced by the same genetic mutation, chromosome 15q partial deletion, and the particular syndrome that will develop depends on whether the mutation is inherited from the child's mother or from their father. This is due to the presence of genomic imprinting in the region. Beckwith-Wiedemann syndrome is also associated with genomic imprinting, often caused by abnormalities in maternal genomic imprinting of a region on chromosome 11.
Transgenerational epigenetic observations.
See main article Transgenerational Epigenetics. In the Overclix study, Marcus Pembry and colleagues observed that the paternal, but not maternal, grandsons of Swedish men who were exposed during predolescence to famine in the 19th century were less likely to die of cardiovascular disease. If food was plentiful, then diabetes mortality in the grandchildren increased, suggesting that this was a transgenerational epigenetic inheritance. The opposite effect was observed for females, the paternal, but not maternal, granddaughters of women who experienced famine while in the womb, and therefore while their eggs were being formed, lived shorter lives on average.
Cancer and developmental abnormalities.
A variety of compounds are considered as epigenetic carcinogens. They result in an increased incidence of tumors, but they do not show mutagen activity. Toxic compounds or pathogens that cause tumors incident to increased regeneration should also be excluded. Examples include diethylstilbestrol, arsenite, hexachlorobenzene, and nickel compounds. Many teratogens exert specific effects on the fetus by epigenetic mechanisms. While epigenetic effects may preserve the effect of a teratogen such as diethylstilbestrol throughout the life of an affected child, the possibility of birth defects resulting from exposure of fathers or in second and succeeding generations of offspring has generally been rejected on theoretical grounds and for lack of evidence. However, a range of male-mediated abnormalities have been demonstrated, and more are likely to exist. FDA label information for Vidasa, a formulation of 5-acetidine, an unmethylatable analog of cytidine that causes hypermethylation when incorporated into DNA, states that men should be advised not to father a child while using the drug, citing evidence in treated male mice of reduced fertility, increased embryo loss, and abnormal embryo development. In rats, endocrine differences were observed in offspring of males exposed to morphine. In mice, second-generation effects of diethylstilbestrol have been described occurring by epigenetic mechanisms. Recent studies have shown that the mixed lineage leukemia, MLL, gene causes leukemia by rearranging and fusing with other genes in different chromosomes, which is a process under epigenetic control. Other investigations have concluded that alterations in histone acetylation and DNA methylation occur in various genes influencing prostate cancer. Gene expression in the prostate can be modulated by nutrition and lifestyle changes. In 2008, the National Institutes of Health announced that $190 million had been earmarked for epigenetics research over the next 5 years. In announcing the funding, government officials noted that epigenetics has the potential to explain mechanisms of aging, human development, and the origins of cancer, heart disease, mental illness, as well as several other conditions. Some investigators, like Randy Epigenetic, Ph.D., of Duke University Medical Center, think epigenetics may ultimately turn out to have a greater role in disease than genetics.
DNA methylation in cancer.
DNA methylation is an important regulator of gene transcription, and a large body of evidence has demonstrated that aberrant DNA methylation is associated with unscheduled gene silencing, and the genes with high levels of 5-methylcytosine in their promoter region are transcriptionally silent. DNA methylation is essential during embryonic development, and in somatic cells, patterns of DNA methylation are in general transmitted to daughter cells with a high fidelity. Aberrant DNA methylation patterns have been associated with a large number of human malignancies and found in two distinct forms, hypermethylation and hypermethylation compared to normal tissue. Hypermethylation is one of the major epigenetic modifications that repress transcription via promoter region of tumor suppressor genes. Hypermethylation typically occurs at CpG islands in the promoter region and is associated with gene inactivation. Global hypermethylation has also been implicated in the development and progression of cancer through different mechanisms.
DNA repair epigenetics in cancer.
Germline, familial, mutations have been identified in 34 different DNA repair genes that cause a high risk of cancer, including, for example, BRCA1 and ATM. These are listed in the article DNA repair deficiency disorder. However, cancers caused by such germline mutations make up only a very small proportion of cancers. For instance, germline mutations cause only 2% to 5% of colon cancer cases. Epigenetic reductions in expression of DNA-repaired genes, however, are very frequent in sporadic, non-germline, cancers, as shown among some representative cancers in the table in this section, while mutations in DNA repair genes in sporadic cancer are very rare. References in the table are given here: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Deficiencies in expression of DNA repair genes cause increased mutation rates. Mutations rates increase in mice defective for mismatched DNA repair genes PMS2, MLH1, MSH2, MSH3 or MSH6, or for DNA repair gene BRCA2, while chromosomal rearrangements and aneuploidy are noted to increase in humans defective in DNA repair gene BLM. Thus, deficiency in DNA repair causes genome instability, and this genome instability is likely the main underlying cause of the genetic alterations leading to cancer. In fact, as indicated by Novak et al. Through a mathematical calculation, the first event in many sporadic neoplasias is a heritable alteration that affects genetic instability, and we note that epigenetic defects in DNA repair are somatically heritable.
Variant histones H2A in cancer.
The histone variants of the H2A family are highly conserved in mammals, playing critical roles in regulating many nuclear processes by altering chromatin structure. One of the key H2A variants, H2AX, marks DNA damage, facilitating the recruitment of DNA repair proteins to restore genomic integrity. Another variant, H2AZ, plays an important role in both gene activation and repression. A high level of H2AZ expression is ubiquitously detected in many cancers and is significantly associated with cellular proliferation and genomic instability. Histone variant macro2A1 is important in the pathogenesis of many types of cancers, for instance in hepatocellular carcinoma.
Cancer treatment.
Current research has shown that epigenetic pharmaceuticals could be a putative replacement or adjuvant therapy for currently accepted treatment methods such as radiation and chemotherapy, or could enhance the effects of these current treatments. it has been shown that the epigenetic control of the proto-oncoregions and the tumor suppressor sequences by conformational changes in histones directly affects the formation and progression of cancer. Epigenetics also has the factor of reversibility, a characteristic that other cancer treatments do not offer. Drug development has focused mainly on histone acetyltransferase, HAT, and histone destylase, HDAC, and has included the introduction to the market of the new pharmaceutical vorinostat, an HDAC inhibitor. HDAC has been shown to play an integral role in the progression of oral squamous cancer. Current frontrunner candidates for new drug targets are histone lysine methyltransferases, KMT, and protein arginine methyltransferases, PRMT.
Twin studies.
Recent studies involving both dizygotic and monozygotic twins have produced some evidence of epigenetic influence in humans. Direct comparisons between identical twins constitute the ideal experimental model for testing environmental epigenetics, because DNA sequence differences that would be abundant in a singleton-based study do not interfere with the analysis. Research has shown that a difference in the environment can produce long-term epigenetic effects, and different developmental monozygotic twin subtypes may be different with respect to their susceptibility to be discordant from an epigenetic point of view. One of the first high-throughput studies of epigenetic differences between monozygotic twins focused in comparing global and locus-specific changes in DNA methylation and histone modifications in a sample of 40 monozygotic twin pairs. In this case, only healthy twin pairs were studied, but a wide range of ages was represented, between 3 and 74 years. One of the major conclusions from this study was that there is an age-dependent accumulation of epigenetic differences between the two siblings of twin pairs. This accumulation suggests the existence of epigenetic drift. A more recent study, where 114 monozygotic twins and 80 dizygotic twins were analyzed for the DNA methylation status of around 6,000 unique genomic regions, concluded that epigenetic similarity at the time of blastocyst splitting may also contribute to phenotypic similarities in monozygotic co-twins. This supports the notion that microenvironment at early stages of embryonic development can be quite important for the establishment of epigenetic marks.
Epigenetics in microorganisms.
Bacteria make widespread use of post-replicative DNA methylation for the epigenetic control of DNA-protein interactions. Bacteria make use of DNA adenine methylation, rather than DNA cytosine methylation, as an epigenetic signal. DNA adenine methylation is important in bacteria virulence in organisms such as Escherichia coli, Salmonella, Vibrio, Yersinia, Haemophilus, and Brucella. In alpha proteobacteria, methylation of adenine regulates the cell cycle and couples gene transcription to DNA replication. In gamma proteobacteria, adenine methylation provides signals for DNA replication, chromosome segregation, mismatch repair, packaging of bacteriophage, transposis activity and regulation of gene expression. The filamentous fungus Neurospora crassa is a prominent model system for understanding the control and function of cytosine methylation. In this organisms, DNA methylation is associated with relics of a genome defense system called RIP, repeat-induced point mutation, and silences gene expression by inhibiting transcription elongation. The yeast prion PSI is generated by a conformational change of a translation termination factor, which is then inherited by daughter cells. This can provide a survival advantage under adverse conditions. This is an example of epigenetic regulation enabling unicellular organisms to respond rapidly to environmental stress. Prions can be viewed as epigenetic agents capable of inducing a phenotypic change without modification of the genome. Direct detection of epigenetic marks in microorganisms is possible with single-molecule real-time sequencing, in which polymerase sensitivity allows for measuring methylation and other modifications as a DNA molecule is being sequenced. Several projects have demonstrated the ability to collect genome-wide epigenetic data in bacteria,